I think we're live. All right. So welcome everybody again to this week's episode of our Actino-based eSeminar. Today it is my great pleasure to introduce Dr. Agustina Undabarrena, who is also a co-host of this whole seminar session. To just introduce her in short, she's from Buenos Aires in Argentina, but did her whole scientific career in Chile, where she first did her studies as a biologist at the Universidad Católica de Chile, and then later had her PhD from the Universidad de Chile in 2017, which she did in the lab of Dr. Michael Sänger and also Dr. Beatriz Camara from the Molecular Biology and Environmental Biotechnology lab at the UTFSM in Valparaiso. And she will talk about NRPS in Rhodococcus today. So with this all set, I guess, Agustina, it's your turn. Enlighten us.
Thank you, Oli, for the kind presentation. Actually, your Spanish is better than my English, so.
Well, thank you.
Thank you. So, as Oli said, my name is Agustina Undabarrena, and today I'm going to talk about Rhodococcus comparative genomics, NRPS distribution, and connection to an orphan metabolite. It's not working. There. Okay. So this is like the final part of the story where you have your paper published and you are jumping in one leg and you're saying, yay, my paper got published. But actually, I'm going to use this scenario to start from the beginning. And I'm going to show you how we end up with two papers of Rhodococcus when actually my PhD thesis was about streptomyces and also my postdoc project was about streptomyces. So this Rhodococcus was always like a side B project of my career. So how do we start? Basically, it was like almost now 10 years ago when Bea started study actinomycetes from the South Pacific. So our lab mainly used three focus. The first one is to study the marine biodiversity. We perform taxonomy and also morphological characterization and to look for the bioactive activity of these strains. Then we perform also genomics to look for these biosynthetic gene clusters in the genome sequences and perform also some BGC networks. And finally, we perform also metabolomics, growing these strains in different media to then perform crude extract extractions, LC high resolution MS, also MS-MS, molecular networking, and trying to elucidate the compounds. So this is our country, Chile, located in the in South America. And you can see that all this is the vast Chilean coast. And over the years, we have been exploring different locations. We have in the northern part, Chañaral Island, also in the insular part, Rapa Nui Island, that maybe you know it from the Moais. This place that was very special for me because was the place where I study in my PhD thesis, Comau Fjord in the northern Patagonia, and also Valparaiso Bay, that is where is our university located. So this actually is our view where in our university. And this is just to show you the marine biodiversity of the actinomycetes in this area. It's truly very different between each location. So this is telling us that it's still worth to go into the depth of the ocean and to retrieve some samples and go to the lab and try to isolate new strains. And Some of you maybe remember that Bea did a talk last year when she actually explained this in depth. This is also the paper that you can look for this in detail. But today I'm going to talk about this special place, the Comau Fjord in northern Chile and Patagonia. And the star of my presentation is this strain, Rhodococcus CA8F. So Rhodococcus CA8F was isolated from Liliwapi Island, from marine sediments, from 22 meters depth. And it was selected mainly because of the bioactivity. So you can see here how the Rhodococcus strain is grown in the middle of the plate, and the model strains that I show you here are grown here, and some of them are completely inhibited. So then we perform a deeper analysis, and we perform culture extraction of this Rhodococcus strain using different solvents, and the ethyl acetate solvent was the one that retrieved most of the bioactivity against these model strains. So this was a strain that has a lot of bioactivity, so this was very interested for us. And then we performed genome sequencing in order to look the biosynthetic gene clusters. We used PacBio, and we retrieved 2 unitics, 1 chromosome, and a linear megaplasmid that, interestingly, it has a lot of genes coding for hypothetical proteins. So Rhodococcus is also very well known for its catabolic metabolism, and we want to explore by genome mining these features. And we found a lot of genes for monoxygenases and deoxygenases. And one of the pathways that is very well studied in our lab is the biphenyl catabolic pathway. So we actually demonstrate that this strain is able to grow on minimal medium with biphenyl as sole carbon source that is very toxic for other strain. And to cope with this stress, this strain also has a lot of genes for oxidative stress response that I show you here. So we have a lot of information about the catabolic metabolism, but what about the anabolic metabolism, right? So our remaining questions of these strains were with such unique genome features, does this strain have also unique biosynthetic gene clusters? In the chemistry part, if these BGCs can be translated into the identification of molecules harboring also novel chemistry. And in the evolutionary part, if we can identify the main mechanism of BGC distribution across the genus when we are comparing our strain with other Rhodococcus. So this is where Paco Barona Gomez came into our life. And he mainly teaches us a lot about using these tools, Bigscape and Corazon. So this mainly you start with a lot of genomes that you can download or you can use from your collections that you put into AntiSmash for BGC prediction. Then you compare it with my big database, and you use Bigscape to actually try to perform a network where you have your BGCs of reference of retrieved from my big, and also your predicted ones. And with this, you try to make correlations. And when you find a GCF or a gene cluster family that is from your interest, then you can also perform Corazon to link this gene cluster diversity to the phylogeny. So that's why was our aim, analyzing the Rhodococcus genus. And for that, we start by downloading all the genomes that were available in RefSec to the date of writing, and that were around 300 genomes. So the first thing that we observe is that these genomes were standing as draft versions. And to perform a comparative genomic study based on BGC networking, these BGC, you must know that are very large pathways, so you need a high quality genomes. And for that, we perform first a pipeline, in-house pipeline of quality controls, applying different bioinformatic criteria and also biological criteria. And we end up filtering these 300 genomes to 110 genomes. And with these 110 genomes, we first did a phylogenomic of the Rhodococcus genus. So I show you here. This is the phylogenomic tree. You can see that there are 4 bigger clades. Clade 1 is here in orange, clade 2 in green, clade 3 in pink, and clade 4 in blue. So our strain that I showed you before from the Cumao Fjord in northern Patagonia is here in clade 4b. And also we wanted to add the isolation source of these strains, so you can find them here in this small square, color square. And the first thing that we observe is that actually there is not a clear correlation regarding isolation source. For example, in the clade 4b, we can see that a lot of these strains come from different isolation source, and there are just 2 marine strains here, our marine strain, and also PR4 that is also from the marine environment. The only exception is clade 2b that, as you can see here, most of them are derived from plants, but still we can see some of aquatic or marine environments. So it's not like a strict correlation regarding the isolation source. And most of these strains groups very divergent in the phylogenomic tree. So next, we perform BGC networking using Bigscape and Anti-Smash. And you can see here that each node is a BGC, and they are connected with each other if they share a row distance below 0.6. Here you can see also the colors that are the BGC's categories. And for example, in blue, there are the NRPSs. That is the most abundant category when you analyze all the BGCs from Rhodococcus. And you can see also in black here, in black label, the BGCs that come from our strain. In terms of network structure, you can see that some of them represent like a really closed balloon shape. And this means that they are very conserved BGCs. And on the contrary, for example, the NRPSs, they show the most diverse pattern of evolution, meaning that these BGCs are still into evolutionary pattern of genetic divergence with gene expansion and recruitment events. And that's why we observe a lot of different BGCs, and they are being shared across all the Rhodococcus. So now when we take this BGC network and we focus only on the NRPS genes, we are making a zoom into the network, and we use colors now to represent the GCF. So when we try to analyze the GCF of the NRPSs in Rhodococcus, we found first that there were 44 GCF, but we couldn't assign a representative or a known representative of this GCF because the score similarities were too low. So this suggests that there is a vast group of NRPSs that is mostly uncharacterized. And in terms also of network structure, we can see 2 patterns. For example, here you can see that traits that are conferring a higher fitness represent a very well-defined GCF. For example, this is GCF corresponding to a siderophore, while traits that are still being tested to fitness, for example, these represent the most diverse pattern. And we can see also an interconnection between GCF1 that is here in orange and GCF5 that is here in green. And you can see how these 2 GCF are highly interconnected between each other. Now, when we take the same NRPS network, but we put colors to represent the phylogeny, you can see that here are the color clades, and some of the NRPS are very specific for certain clades. For example, this that I showed you before, that was a siderophore, is strictly observed in clade 4b, while other ones are being representative in different clades. So we found overall a strong correlation with phylogenomic clades rather than with isolation source, as I showed you before. And this was the first evidence suggesting that maybe these VGCs are being vertically transferred through the Rhodococcus phylogeny. Okay, now we made a heat map where we show the presence, absence of each GCF. So you can see here, there are all the GCF that I showed you before, the 44. And here you can see the phylogeny. So we have the strains, the clades, and also the isolation source in here. Our strain is located here in clade 4B. And this is the connection that I showed you before between GCF1 and GCF5. And you can see a really interesting pattern that when one is present, the other one is absent. And so is in the opposite scenario. When one is present, the other one is absent. So we want to see a little bit in depth this kind of presence as absent correlation between GCF1 and GCS5. And to do so, we study one NRPS of our strain that has a representative within GCF1. And this is the NRPS5 that I show you here. So this is NRPS5, and it's composed with a left arm, middle section, and right arm. And you can see in blue, there are the core biosynthetic genes. In light blue, the additional biosynthetic genes for tailoring reactions. In purple, the genes from transport-related genes. And in green, also the regulatory genes. And I'm going to explain this cluster in depth, taking first the left arm, then the middle section, and then the right arm. So we use Corazon also to see how was the distribution of this BGC. And for example, in the left arm, we could observe that all these genes had predicted to be involved in the synthesis of a C4 molecule and a modification of this molecule. And we grouped these genes based on this function. And when the genes have a function together, we put here a geometric symbol. For example, this is the case. These two genes are transporters. These two genes are regulatory elements. This is the kinase, and this is the transcriptional regulator. And all these genes are working together in the synthesis of the C4 molecule. So if the genes are working together, then we put a geometric symbol and we group this gene as a biosynthetic block. In the middle section, we have three main NRPS genes. And you can see here the prediction, the anti-smash prediction. This is 17 amino acids, but most of them are unknown. And also, this cluster has some unique features, like this is a C-starter domain in the first NRPS gene, and this is a standalone adenylation domain in the third NRPS gene, that altogether add a little bit more of divergence into the cluster. And in the right arm, we perform the same analysis using Corazon, grouping the genes into biosynthetic units. And you can see how these genes are all involved in the synthesis of a chloramphenicol-like molecule compound that has also a modification of a nitro group, mainly given by this gene, paraminobenzoate N-oxygenase, or F, that is here in yellow. So having the genetic in mind, we wanted to see whether this VGC was distributed across phylogeny. And for that, you can see here in the upper side of the figure, this is all the cluster that I showed you before of our strain. And we compare this distribution with clade 4A and clade 4B, where our strain is located here. So where you can observe that the geometric symbol has the color, it means that all the genes that are composing that biosynthetic unit are present. And when you observe that this geometric symbol is white, that means that at least one of the genes are missing. And we use this legend to make a comparison of the VGC across all the strain, and we perform this figure that is here. And you can see that, for example, when you found these two lines, it means that it's found in another part of the genome, meaning that it's not actually part of the cluster. And that's happening in clade 4A, that you can see that the middle section is mainly composed of one NRPS gene, not three, and it's close to the right arm, but only this part of the right arm, that is the triangle, so you can only find these 4 genes, and the others are somewhere else in the genome, and the left arm, it's also somewhere else in the genome, and this is not like the classic configuration of the VGC as we observed in our strain. And in clade 4B, we found that the middle section now is not conserved as in 4A. The middle section, we found that we have sometimes 3 NRPS genes, and sometimes we found only 1 monomodular NRPS gene, as you observe here. And this is the most divergence part of the cluster. And then we see the left arm and the right arm, and same as before, the left arm is not always located contiguously, the middle section and the right arm. And the right arm, it's not always fully present. Like you can see here that, for example, these genes are present, but in these genes that are represented from these 2 genes, they are mostly absent. And this is in terms of genes, but now we wanted to see if also we see this difference in terms of modularity. So you can observe here all the strains that have 3 NRPS genes, and you can observe here that is our strain. And in terms of NRPS modularity, we can also see differences. We can see that, for example, this strain that is our strain actually has that is the adenylation standalone domain, but also it has 1 module more than comparing to this strain that is the PR4 that comes also from the marine environment. So when we compare in terms of genes and also in terms of modularity, we can observe that there is no such unique VGC, no such identical VGC in the other strain, and our strain harbors a unique VGC. Also, it's interesting to observe here that maybe this adenylation standalone domain has arise from a duplication of this adenylation domain, because we observe here in these 3 strains that are farther away in the phylogeny, that here you can find the complete module, and maybe this was a duplication, or maybe the other way around, it can be also a loss of this adenylation domain, and that's why you observe these different configurations in these closely related strains and these far away strains. Okay, so that was in terms of the genes, and now we want to explore in terms of the chemistry, and for that we perform the OSMAC approach, that is the 1 strain many compounds. So we grow this strain in different media that I'll show you here, and then we perform ethyl acetate crude extracts and bioactivity assays against some of model bacteria that I show you here. And we select the 3 most bioactive extract for the replication using LC high resolution MS in collaboration with Fundación Medina. So Medina retrieved this result to us. So this is the LC high resolution MS, and we can observe 3 different peaks that I'm showing you here, the UV profile and the MS profile. And when Medina compared these profiles with the in-house database, For compound identification, they found that these compounds were coreinosines, coreinesin 1, coreinesin 2, and coreinesin 3. So coreinosins were first reported in Corynebacterium, but they were not so far described in Rhodococcus. So actually, this is the first report that tells that coreinosin is being detected in a crude extract from Rhodococcus. So as a graphical abstract, I show you that we have this strain from the Kumau Fjord. We perform genome sequencing and some comparative genomics and phylogeny to perform BGC similarity networks using BigScape. Also, we did genome mining to look for the biosynthetic gene clusters in our strain, and we found this very interesting NRPS5 mega cluster composed by a left arm, middle section, and a right arm that is very unique across the phylogeny. And also we use Corazon to detect that, and then we perform some crude extracts, the replications with Medina to find the coreinosins that are the molecules structurally related to the chloramphenicol. So in the genomic part, we perform a comparative genomic pipeline for analyzing BGCs. We group NRPSs into GCF, and we reveal that most are uncharacterized families. We also correlate the NRPS pathways with phylogeny, and we could observe that they are vertically distributed. In the chemistry part, we found this NRPS5 that is a mega biosynthetic gene cluster composed by a left arm, middle section, and right arm. And the right arm is involved in the synthesis of a chloramphenicol-related molecule, as I showed you before, the coreinosins that were first described in Rhodococcus in this report. And coreinosins present bioactivity in Rhodococcus crude extracts. In the evolution part, we show you that NRPS5 is unique within this clade using Corazon, and that the left arm and the right arm are the conserved along for clade 4b, but the variable region is given by the NRPS genes, resulting in a very variable peptidic core and also variable modules. So the genomic, the chemistry, and the evolution BGC patterns of these strains present some novelty. And we have still some remaining questions. So in one part, we are trying to elucidate the biosynthesis because, as I showed you before, coreinosins is the product of the right arm, but also here the middle section is assembling a peptidic core and the left arm a C4 molecule. So we are trying to find out how they are all assembled all together and how maybe we can see this as the final product of the biosynthesis. And in terms of structure, of course, we want to purify and make also structural elucidation of the respective products of the clusters. Hopefully, we can see this final product of the cluster M4 molecule. So if you're interested in collaborate with us, just write to us. This is Bea's mail, and you can write to her if you are interested in performing one of these still remaining questions that we have so far. So with that being said, I just want to say thank you. This is a photo of my Actino team before the pandemics when we were all together. This is Bea that I have to say thank you for supporting me all the time. Also Andres, Leo, and Richie that were truly active collaborating with me in this paper, and Paco and Lalo that were also collaborating with us in this story. And thank you for listening. And here's my mail if you have further questions, but I will be very happy to answer some of them now if you have. Thank you so much. Back to you, Ali.
All right. Thank you, Agustina, for this fascinating talk. Really nicely delivered from start to finish. Since there are no questions yet, it is my privilege, of course, as a chair to have the first one. So in having the connection, so you still want to go for the connection of the biosynthesis of those compounds and your gene cluster. So are you planning to do the more or less classic approach, trying to knock out parts of the gene cluster and then see, do I still have bioactivity, or do I, if you knock out just the parts yet that you don't know what they do yet, and then see, will you even still have the bioactivity of the right arm it was, right, from those Bacterium, yes. So is this something you are planning to do?
Yes, I'm trying to go to this previous slide, but I can't wait. Okay. Here, you can see it, right, Oli?
Yes.
Yeah, okay, yes. So actually what I show you is that we retrieve the compound from this part of the cluster, right? This is the part of the cluster that it's involved in producing the chorinosins. And that's what we observed in our crude extracts. So this crude extract actually has bioactivity. So now we want to see, of course, how this is interacting with the middle section and with the left arm, that it's also involved in producing a C4 molecule. And we believe that this is the first monomer, because here we have C starter that has the ability to incorporate this molecule into the assembly line. And for this, yes, we are planning to perform several knockouts into the pathway, and then to see if we can retrieve different molecules. But one of our biggest limitations is that this final product that we believe it's the final product is actually a very big one, because you have here 17 amino acids, and then the chorinosins, and then this molecule. So actually we have like a chemical limitation to detect like the, the biggest product of the pathway. But if we can like solve that, yes, we are planning to, like, our ideal is to have okay, this is like a genetic part of the phylogeny of clade 4b, and we want to have here, like next to it, the chemical part, no? And we want to see how all these changes in gene changes are also affecting the structural part of the chemistry. And we want to see how this is also observed in this clade, because we believe that if this biosynthetic gene cluster is unique, then we will have also a unique molecule. But also we observed these very small changes in genes along the clade. So we want to observe if that changes are also a product of the structural changes that we can observe in the molecules. And we, of course, want to elucidate the bioactivity to see which one of them are the most bioactive ones. But yes, that's our part of the future work. And that's why I put that if you're interested in helping us or collaborating, please write to Bea. I think she needs more hands to do all the mutants that she needs to, to elucidate the part of the pathway. So that's why I leave the message there for everyone that is interested. But we will be very happy to perform the mutants.
Right.
Actually, I begin that part. I went to Lorena Fernandez Martinez lab in Edge Hill University. And I tried to use her meganuclease approach to try to knock out some of these genes. And unfortunately, I was only about two months and a half, I think. So it was not enough time to perform all the mutants that I needed yet. But I think we are in the process of achieving that, hopefully, in the future.
All right, looking forward to your next talk then.
But
sorry?
yeah, but I don't know if I'm going to do that because I have streptomyces all over also. This is like my side project, Rhodococcus, but still, yeah.
Well, that's a quite impressive side project then. Okay, so letting somebody else speak. David Mark says, fabulous stuff. Have you checked to see if Corynebacteria and your Rhodococcus share any clusters?
Yes, we use also corazon to, in comparison with the Corynebacteria that are available, the genome sequence that are available, and we couldn't find our cluster in Corynebacterium. So we actually believe that corynesians were detected in a Corynebacterium that was not taxonomically proper identified, and we believe that maybe that strain was a Rhodococcus and not a Corynebacterium because we couldn't find our VGC or any similar VGC in Corynebacterium.
Okay. And also, did you have a actual reference to the actual strain where those corynesians have been found? So did you have a genome of this specific strain that apparently produced the corynesians? Yeah, okay, then in this case, it's of course hard.
No, because that strain was, there is only one reference. That is a very old paper, like a paper from the 70s, that they were actually only demonstrating that they elucidate the compound, the corynesians. And they, of course, at that time, they didn't have the genome sequence. And
Yes.
that's the only reference that we have so far. And that's why we think that maybe the Corynebacterium was misclassified, because at that time also the taxonomic identification was mainly morphological classification. And Corynebacterium and Rhodococcus sometimes look very similar. So that's why we believe that maybe it was classified as Corynebacterium, but maybe that strain, the original strain, is a Rhodococcus. It's very interesting, but we don't have the strain, and also we don't have the genome, so we cannot like demonstrate it, but that's our guess.
Yeah, sounds like a reasonable, educated guess. Okay, so Kate Duncan says, hi, Agustina, lovely talk. You mentioned in relation to NRPS 5.
Yes.
You can you comment more broadly if the location of the BGCs in the genome is conserved and if there's any evidence of genomic islands?
Yes, nice question, Kate. Thank you for that. I cannot go, wait. Yes, here. So we try to understand how this cluster was conserved along the clade 4B. And mainly, we didn't find any evidence of a genomic island. So that's why we also believe that the distribution is a vertical distribution and not a horizontal distribution of the cluster. We found that every single strain of this clade 4B has at least a representative of this GCF, that is the GCF1 or the GCF5. But when we observe the biosynthetic gene cluster is where we observe actually the differences and also in modularity. And for example, here you can see that mainly the left arm is located somewhere else in the genome, not always like close together with the middle section and the right arm, meaning that the genes are present, but they are not actually constituting a cluster. And when they are, and these are the strains that I show you here, that these are the clusters that are like the most similar clusters, that are these strains and this strain and this strain, they observe the difference in terms of modularity. So this means that all these strains, even with that they have 3 genes, they have different amino acid, and also the incorporation in numbers and also in terms of the amino acid that they are incorporating is different. So that's why we believe that the chemical part of the cluster, it's also going to be really different. And we don't observe a cluster similar to the one that our strain has, that is right here, even in strains that are very similar in taxonomy. So that's why we believe that we have a very unique cluster. And also that's why we believe that the compound is also really unique.
Okay. Since there are no more questions right now, if you are okay with it, I would have another shot actually at the part with about the biosynthesis and the biological activity.
Yeah.
Since you showed us those samples that have been measured in high resolution MS in Spain, was there a was this the picture you showed us, the chromatogram? Was this a from a fraction that was actually measured, or was this a complete extract that was measured?
It was the crude extract from the bioactive crude extract here from the ISP2 media, the ones that have all this activity.
Mm-hmm.
Yes, so it's all the crude extract, but they only found 3 peaks. So we didn't have to purify it a lot. But the only thing that they observed in that crude extract was coreinicins, because peak 1 is this coreinicin 1, peak 2 is coreinicin 2, and peak 3 is coreinicin 3. So actually there is no other molecules in that crude extract.
Okay, would have been interesting if the bioactivity you see is definitely just from those 3 compounds, or maybe if you were to test if we were to prepare fractions, if you maybe see something in another fraction, so maybe actually already see already have your final compound somewhere in there. But okay, of course, if you already have, if that's the whole chromatogram.
Yeah, that's why I was telling you that we have like a chemical limit of detection, because our final compound it's actually really big, and that's why we believe we are seeing just a part of it and not the bigger part. Maybe it's there, but we are not actually seeing because of the limit of detection.
Okay. Great stuff. Also, Kate Duncan wants to again thank you, and wanted to ask if those other GCFs they are fairly conserved, and are there similar genome? Are they in similar genome locations too?
Which one? Sorry.
The other GCF families.
The other GCF. I am not sure what she means.
The genetic cluster families, or what's they're called again? If you go back to the page with the clusters that you showed us.
This one? These are the GCF.
Yes, and the question was if those other ones are also conserved, and if they are in similar genome locations as well.
The most conserved GCF that we found was this one, GCF9. All these clusters are involved in the synthesis of a siderophore, as I told you before. Yeah, right. Yes, and this was the most conserved that we found. The others were kind of messy, and that's why we believe that they are like in a dynamic pattern of distribution still, and the products are being tested to fitness, and that's why the genes are all being shared. And when we believe that when the trait has confer to the strain a higher fitness, like for example a siderophore that is very important for the survivance of the strain, then we can find like a very close pattern of distribution, like the one that we observe here, like a balloon shape, really, really close pattern of distribution. So I think that like the dynamic part of the evolution, it's where the most interesting part is, because those genes are all being shared and they are being tested to fitness. And if they are conferring a trait that is important for the strain, then they will like go to performing this closed pattern of distribution. But we are still like seeing this picture, and we are still seeing like right now in this evolutionary frame, they are all in this dynamic process. So that's why we are very interested in looking in these biosynthetic gene clusters, because they are all being very not the conserved ones, but also the shared ones between the strains. And those products are the ones that are being tested. So we are interested into maybe have one of those products to actually see if we can correlate the vast chemistry with also the vast genomic divergence that we are observing in this GCF.
All right, I guess this answers the question. At least there was no further asking. Yes, that's all the questions. So,
well, if you have any more questions, you can just write me. I put my mail at the end of the talk. So you can always write me if you have any more questions. I will be very happy to receive your feedback and suggestions. I will put my mail there. Yeah.
All right, you can see it right there, guys. So, Agustina, thank you again a lot for this great session today. And to all the ones who are still listening, I want to remind you that at the 20th of October, in 2 weeks from now, Kai Blin will talk about anti-smash and what you can do with it right now. And I think you should have already received a way to by mail
Agustina-based mail.
Yes, exactly. Thank you, Agustina. to actually send in questions, because Kai would like to make kind of a bit of a workshop and be prepared for some actual questions from users, so he can actually answer those and give helpful information for everybody using his program. So, and in case you have not received it, we will also release on Twitter the contact information for him for his talk. So until then, I guess it's goodbye, and see you all in 2 weeks.
Thank you, Oli. Thank you, Molly.